So thanks to the Element team for inviting me to come speak about a collaboration we've been working on with them. Started discussions even a few years ago with Molly in the early stages, and so it's been really very, very wonderful collaboration to take some of these very unique and precious samples that come from astronauts, or in some cases other crew members, to get them to come up. Actually, I can see what's on stage here. There we go, great. This one just keeps changing over here, but now it's fine. Okay, great. So really wanted to take an application of the element and AVITI chemistry to unusually rare samples, things that come from spaceflight. So in particular, we've been working with NASA and SpaceX on taking a lot of the tools of modern molecular medicine and precision medicine to apply them to what we see in some of these crew members as they go to and from space stations, which I'll tell you a bit about some of this work that we've done to look with these crews. So in particular, some of the very first work we did was with NASA and the NASA Twin Study to see what happens to the body when you're up in space for a year. This is some beautiful pictures that Scott Kelly took when he was up in flight. You can see here, for example, also images of our planet as he's taking these shots and would post them up on Instagram. But one of the things we think about is when you're up in space, you're really enduring any, say, new clinical therapy, any CAR T therapy, anything that's happening to your body. There's a continual shedding of cell-free DNA, which has been discussed a lot at this meeting, that happens in the body. And we know this is mainly through apoptosis, necrosis, and secretion, but this actually gives you really a whole body molecular scan as you look at DNA as it's kind of moving throughout your body, as well as things that are packaged in exosomes that also are informative. And so it's informative because it tells you which cells are undergoing stress, which ones are dying, which ones are really undergoing any sort of trauma. And this is true, again, for anything happening on Earth, but also anything up in space. And if you look at the way the reads map back to the genome, they also tell you which areas of the chromatin in cells are open or closed. So in particular, you can see different mapping of where you see nucleosome depletion, where you see these drops. You can actually see those are actually areas of the genome that are very active. You're seeing profound gene expression changes. And if you see it staying relatively flat, those are areas of closed chromatin. So the mapping itself of cell-free DNA, when you bring it back to the genome, it tells you what is the activity of different cell types in the body. And so it gives you a chance to just using cell-free DNA alone give you a way to measure, if you will, what Trevor Noah once called space genes. So it lets you kind of see what they are. And the way we originally did this with the twin study was actually looking at a lot of the omes, looking at microbiome, metagenome, we're looking at telomere changes, and metabolomics, looking at every modality of an expression change and molecular change that we could. But we've since expanded this to be more broad. So it's not just NASA astronauts, it can now be anyone. We've expanded the IRB to include really any commercial spaceflight as well, which is its own burgeoning economic sector. And so this is a new protocol that's called the Standard Omics Measures for Astronauts, or what's called SOMA. And this includes a lot of the same metrics we've done before for other astronauts, but now is focusing a bit more on using really high quality sequencing, in particular what we've been seeing with element data, to start to look at all these other layers of biology, but then also look at cell-free DNA, which I'll tell you today a little bit about, as well as some of the other microbiome and single cell gene expression and chromatin changes. So you can see here in this picture, my student couldn't resist coloring stool brown in this image, but that was, and urine yellow, but the other colors are just by random choice. But in this case, we got the very first chance to try this protocol actually last year. It was in September, was when the Inspiration 4 crew went up for the first all-civilian crew up into flight, also the first cancer survivor, and also the first prosthetic limb flown up into space with Haley Arsenault over there on the right. And so this was a really extraordinary event. Actually, if you've never been to go see a launch, I highly recommend it. Actually, you can see here at Cape Canaveral, you can see in the distance the launch pad. It gets loaded up with fuel. You can see actually as the launch happens, you can actually feel it on the ground as the sound waves move through the ground faster than it moves through the air. You can see the Dragon rocket, the capsule making its way up into space, and you can see the contrails kind of come back. And the Inspiration 4 mission actually went up to almost 600 kilometers. It was higher than the Hubble Space Telescope, higher than the space station. So it floated up there for 3.5 days, and eventually they came back down. This is right before they landed, and then also splashed down, and they got picked up there. And they were very excited when they landed back on Earth because they didn't die when they came back. We were very excited because they had lots of precious samples, and we were glad that they also didn't die. We're also drinking fireballs here. And then the postdoc who met with the SpaceX crew as they brought the crew back, and this is a capsule once it gets back onto the loading dock, and also get HEPA filters and other samples. And so we're excited because this is the beginning of a real burgeoning, as I said, commercial spaceflight sector, where more and more people will be heading up into space But as they do, we really want to understand what happens to the body. Could we use some of these tools like cell-free DNA profiling and multi-omics measurements to better understand the risk to the body and to prepare for eventually longer missions? So in some of these discussions I'd had with Molly and Sean and others and Mike at Element, we thought, well, you know, this is a platform that ostensibly sounds like it's going to be, maybe best in class in accuracy, and sounds like it was going to be really great. So we thought, well, let's see if we could use this new chemistry, this new platform, to understand these samples before and after spaceflight for this crew and a plan for future crews. So one of the first things we did, as we do with almost all data when you get back to the lab, is we say, okay, let's map it to the genome and see what the quality looks like. And in this regard, we could actually see the quality looks extraordinarily good, all the way out to 150 nucleotides. Actually, just before this talk, I was asking Molly, I said, how far could you take it? Because I think this looks fabulous. You could really probably push it out to 200, 250, 5,000, I'll say. Since I'm on stage, I'll say go to 5,000. That's not official. I just think you should see how far it can go. But it looks really good. The quality, it looked very good. It still does for all the runs that we've done. Also, the mapping quality was extremely good. So we're seeing, most of the reads mapping well above 99% for our mapped reads. Not that many unmapped reads or multi-mapped reads, and the read length also looking good. So as I showed you in that earlier plot to see where the reads map across different structures across the genome, this is for pre and post flight, we want to see if you infer where they map across the genome, you can actually infer what cell type they are. So this is some of the very first data I was getting last night, is to see what cell types do we see. You can kind of see it on top there. Post-flight on the left and then pre-flight on the right, you can infer which cell types are dying and shedding their DNA into the bloodstream. And you can see natural killer cells, monocytes, CD4 and CD8 cells, T cells and B cells up at the top, which is what you'd expect. These are cells that are in the bloodstream. This is very much what you think are cells that would normally be shedding. But we do see differences post-flight. We see actually a little bit more of some of the CD8 cells and B cells are being shed, or maybe potentially even from liver and pancreas starting to show up as more of a signature, which indicates there's some stress on these organs. You can actually use this data to infer which organs are undergoing the most stress during spaceflight, or again, it could be during anything, any sort of clinical treatment, having hot sauce, it could be anything. But this kind of shows that we're able to use this data quite well to infer that. And also we saw that the inserts, the fragment length of the sequences were relatively consistent though. As many of you know, these fragment sizes are different in cancer, and so there's another indicator of if something happening in the body that might be indicative of stress. In this case, they actually are very stable, which is a good sign, because the mission in this case was only 3 days of an orbital flight and relatively stable, so we were glad to see this. So that was some of the data on cell-free DNA, but we can use the sequencer for anything. It's a sequencer. So of course, then we also want to look at some of the latest protocols from single-cell profiling using the multi-ohm kits. And here we looked at pre and post-flight using the ATAC-seq and gene expression multi-ohm kits. In this regard, most of you are probably familiar with this protocol, but this is basically where you can do nuclei isolation and then also look at chromatin accessibility as well as gene expression from these cells. And then once you get the data, you can actually finish the libraries, align to the genome, call peaks, and also do gene expression quantification, and combine these to do joint feature linkage for these cells. And what we could see is actually we wanted to take basically the same samples, run them on Illumina, and run them on Element, and see how do they compare, because ideally this is something we want to really benchmark. And we could actually see, if you look at the alignment summary, we're seeing the number of peaks, the fraction of the genome in the peaks, and the confidently mapped regions, and you can see across these samples, they looked very consistent. If you look at, say, you can see Illumina, and then some of the same libraries run on Element done in replicates, and you're seeing it looks almost indistinguishable. In some cases, we're actually seeing more high-quality fragments mapping actually to the genome, so we're seeing some cases a little bit better data showing up on Element, which is good to see. And then we wanted to look at, okay, that's just mapping. If you actually then call peaks, and then you call expression changes, what do you pick up? In this regard, we could see that the ideal profiles up on the left, and you can see for Illumina and Element, they look nearly identical. You can see these profiles of what you see for the targeting plot, that these also look pretty indistinguishable. And these are all, again, QC metrics, but they look about the same. Then we look for transcriptional start site enrichment. And again, here the point is that these plots look almost identical, so we can see very, very comparable data between both systems, and they look ideal for TSS data. And then again here also looking at what's the insert size distributions for the nucleosome profiles from ATAC-seq data. And here you want to see this periodicity to show that they're actually seeing accessibility of different chromatin. And here again, they look very similar. So at that point, we just said, okay, this data looks really good. The quality is really essentially indistinguishable, and in some cases better than what we're seeing for Illumina data. So we said, okay, what's changing in this crew? It's only a relatively short mission, but we want to see which cell types are changing. And we see here it's B cells and T cells and monocyte populations that are the ones that are the most dramatically changing, and we can see in terms of their proportion. But then we wanted to look at, actually, this is one of my favorite parts of the multi-ohm kits, is you can then see which cell types are changing. You can see this top plot is by colors up and down expression in orange and in purple. Genes that go up and go down, and each vertical line is one of those genes. In each of the cell types are the horizontal lines. So you can see many, many thousands of differentially expressed genes that changes. That's not surprising. Genes are changing constantly in our bodies. But the differentially accessible regions, those sort of DARs, these areas where chromatin is changing its structure in response to spaceflight, as you can see, the monocyte population is much more profoundly impacted by going to space and then coming back versus the other cell types, which are just changing sort of the light switches of their gene expression rather than rearranging the electrical circuits, if you will. And so that was another application which looked really good. Then, of course, we wanted to take, as another application, look at microbiome and see what happens to the crew. Do they start to become more similar? Kind of what we see, for example, when roller derby. If you play roller derby, your skin microbiomes will get more similar to each other. So when the Celtics tonight might become more like the Golden State Warriors, for example, that will probably happen during this game. Some people think it's totally gross to think about. I think it's totally fabulous to think about that kind of exchange. But in any case, to this question, if you're going to a small capsule for a few days, will your microbiome look more like your crew members? And so to do this, we looked at 10 different areas of the body and swabbed and sequenced them. And here we could actually see that indeed during the course of the mission, what species changed. We could see highlighted in red. There were many, many species that changed slightly, but some of them statistically significant did change. And you can actually see here which particular species changed. It was in this case a particular of Caulobacter, which are gram-negative oligotrophs, that did change and actually did get a little bit more similar to each other during the mission. So also the toilet broke on the Dragon capsule for this mission, so they also had to wear a lot of diapers, which changed their microbial profile on their skin in ways I won't go into details with right now. But in any case, what's happening next? We've really enjoyed the partnership with Element. The data quality is, in every instance we've seen, as good or better than what we're seeing for sort of the mapping and the functional genomics approaches of these data. So we're very excited to apply to other missions that are upcoming. Actually, the crew for the Axiom 2 mission has come into the lab recently to do training, and so we're planning to do some of the same assays as well as new ones with these crews. Eventually, you could imagine taking it for crews that go up longer in space. There's going to be, if all goes according to plan, 4 different space stations in orbit around our planet over the next 3 or 4 years, so really exciting time for the low Earth orbit. And eventually there'll be this system of the Polaris, the Polaris missions, or the Starship rocket that will go to the moon, eventually then go to Mars. Some of the first crews there, like the Polaris Dawn crew, will also go out in flight. And so we'll start to, hopefully the Element system by then will be deployed maybe on the moon base at some point. There's early discussions, I think, on this. And then eventually put the system on Mars. I think you should do it there, again, by 2035. So it's a little bit future planning, I'd say. But in the end, I want to thank everyone in the lab who's really made this work possible, has worked really long nights, including last night to get some slides to me. And then also, in many nights we're staring at data, I kind of feel like you're looking like this at the screen, but really they make this possible, and these people are awesome. I couldn't stand up here without them. Thanks again to funding, of course, to the Gates Foundation, World Quant, the NCI, and NASA, and of course, many, many friends, colleagues, and collaborators. And finally, of course, thanks so much to Element for this really fantastic collaboration and chance to work with you all. And thank you very much.